Okay, let's get started. So we are reaching the end here of the semester. I can't believe that time has gone by that fast. Okay, so we have today's lecture and 2 more next week, and we'll be done. You might have noticed that I've sent you an invite for a review session for Wednesday. So there won't be lab next week. So there will be a review session instead, but only one on Wednesday during lab time instead of having Monday and Wednesday. So we'll meet all together as a group one time on Wednesday. What I'll be doing is actually, I might spend the first 20, 30 minutes finishing up lecture material so that if we finish review, we will cancel lecture time on Wednesday. So we will end when the review session ends. So that's kind of my plan. We'll see how it goes depending on how it goes on Monday, and I'll see how much lecture time I need. If I need only a little bit of lecture time, I'll just cover it during the review session and then start reviewing. One thing I want to remind you is I have posted on Canvas a set of review questions. So you'll find them under week 15. So I encourage all of you over the weekend to start solving these questions. During the review session, I will go over each question with you, each of these review questions. I will answer any questions you have regarding any topic for the that we covered in the semester. And I guarantee you that some of these questions will be on the final. So you want to make sure you solve these questions, and you want to make sure you're there during the review session so that you don't miss out. The final is 200 points. So although you have a lot of points on labs and other parts of the class, but the final is a significant component. So I'd like you to be very well prepared. Come ready with questions for me too. So the more questions you have, the more grounds we can cover in terms of reviewing the material together. Yeah, so that's what I have for you.
About the final, how is it going to be like the same format as exam 1, I guess, for like the types of questions, like short answer?
Yes. And also it's going to be the same format as the review questions I posted on Canvas.
And is it going to cover material from the whole semester or just what was not covered on exam 1?
So it is cumulative, and the reason is a lot of the material we covered in the first part of the semester show up throughout. So like chromatography, for example, it's integral to a lot of other topics we talk about. You want to know sample preparation because it's important for everything we talk about and the entire semester. But I can tell you that the main questions will focus on material after exam or midterm 1. So the questions will be focused on the material after the midterm, but there are concepts that you really need to know that we covered at the beginning of the semester. So does that make sense? Chromatography is integral.
Okay.
Okay. Any other questions regarding the final? Okay. All right, so today's topic is immunoassays. So we'll go over some basics of immunoassays and some also applications. So when you hear immunoassay, what does it mean to you? How can you describe immunoassays? By few words. You can type, unmute yourself, whatever you want. What are these kind of assays, immunoassays? I'm sure you came across them. Have to do with allergens, ELISA, proteins, antigens. Yes, so we're getting a few words here and there. Use of antibodies. Yes, antibodies and antigens. Exactly. So immunoassays involve antibodies, and antigens are whatever components that would react to the antibodies, mostly proteins. But there are other antigens that we can also detect using antibodies. So mycotoxins, for example, antibiotics, pesticides. So types of toxins like aflatoxin. So mostly it's proteins, but there are also other components, small molecular weight components that we can detect using antibodies. So antibody reacting with antigen is the basis of the immunoassay. And antibodies are proteins themselves too. An antigen can be a protein, like an allergen, for example. If we want to determine allergenicity of a certain protein, we can see if that protein reacts with a specific antibody and give you an immunoreaction. But like I said, it is not only the antigen cannot only be protein, can be protein or any other components. So since immunoassays involve antibodies, we're going to talk about antibodies a little bit. So this is a very common antibody shape or the composition of the antibody. So it's basically made up of 4 protein polymers. So you have the heavy chain, which is the blue here, 2 identical heavy chains, and 2 identical light chains here. They form kind of a Y shape. And your antigen binds to this area. So you have 2 binding sites on every antibody. So that's the binding site here and another binding site, and this is the green is the antigen. So antibodies are produced by specific types of cells in our bodies, the human bodies and animal bodies, and they call them the B cells. So that when an antigen or a foreign body is introduced in our system, B cells respond by producing antibodies. And these antibodies would capture the antigen and try to get rid of it. So the interaction between the antigen and antibody are very, very specific. And they are very strong interaction, although they're non-covalent. So it's not a covalent interaction. It could be hydrogen bond, hydrophobic, electrostatic, van der Waals, but not a covalent interaction. But it's very, very specific. It's kind of like a lock and key, or kind of like similar to enzyme substrate interaction. It's very specific, among the strongest interactions between molecules. This is an IgG or immunoglobulin G kind of a structure. Again, you see that Y shape here. And like I said, the affinity is really high between the antibody and the antigen. And like I said, the interactions are ionic, hydrogen bond, hydrophobic, van der Waals, but not covalent. So you will hear me say epitopes quite a bit. So an epitope is a region on the antigen. If we think about it as a protein as an antigen, you have regions on the protein that can bind to the antibody. That particular region on the protein is called epitope. So you will hear sometimes linear epitope or conformational epitope. Linear epitope is like here. So you have amino acids in a region here that are in a sequence, 1, 2, 3, 4, 5. That could be an epitope or a binding site for the antigen, for the antibody. When we say conformational, if it's a protein, it's going to fold, right? A protein is a molecule that folds. So the way that molecule folds can form a region due to folding. That region is the region that binds to the antibody. So that makes it, this form makes it conformational antibody. So linear and conformational. It's a small region. It has a size of about 7 glucose units. And the one thing that I want to have you pay attention to is the type of antibodies. So you have antibodies that are polyclonal, and you have antibodies that are monoclonal. What does that mean to you? So the monoclonal antibodies are basically antibodies that recognize the same epitope, only one epitope. However, the polyclonal, here I discovered how to write on my screen or draw. Okay, so the polyclonal is a combination of antibodies that might recognize different epitopes on the same molecule, same protein. So let's say here I have a protein. You know, that is a structure of a protein. You think here I have an epitope. So I have an antibody that basically react with this position. That's an antibody reacting with one epitope here. And I have another antibody that will react here. So these are two different antibodies. They react at different places on that protein. So basically this is epitope 1, this is epitope 2. So this makes it polyclonal antibodies. They are not the same. Monoclonal means that they are antibodies that can only bind to one epitope, one type of epitope. So this is important because monoclonal are standard kind of reagents. So they are antibodies that are, you know, in assays that we purchase. So they can be produced and they're helpful when we're running ELISA, enzyme-linked immunosorbent assay. So when we're running ELISA, we're using standard antibodies. They're all monoclonal. They bind to one specific epitope. However, if let's say we inject a certain protein antigen into an animal's body, the body will start an immune response, will produce antibodies. The antibodies are coming from many different B cells, so they can interact with this protein in many different locations. So these will be, if we collect the serum, that would be a polyclonal serum containing polyclonal antibodies. So how do we produce monoclonal? So there was like, I don't know, 30, 40 years ago, they discovered that if they get one type of B cell and they fuse it with a cancerous cell, so then they can culture it. So they can produce more of these B cells, the same type of B cells, and the same type of B cells will produce the same type of antibody. So then you can produce these monoclonal antibody for preparation of assays and acting as standard reagents. It was pretty neat. So immunoassays, these are just general points of immunoassays. They are specific, they are sensitive, and simple. So like many times you get these reagents in an assay and you just run a simple assay that is based on measuring absorption. So we really have to have specific antibody for what we're trying to measure, which is the antigen of interest. And we should be able to separate and differentiate the bound antigen from a free antigen form. And how we do that is by labeling. And I will talk about labeling in a minute here. There is a common feature in proteins that is very useful in these assays is their hydrophobic surfaces. It's because of their hydrophobic surfaces, they can bind on our plate, the plate that is made up, that is basically has a hydrophobic surface. So the binding of these proteins to the plate or binding of the antibodies to the plate is very essential component of the immunoassays. And the binding is via hydrophobic interactions. Now, how do we produce antibodies specific for measuring a target antigen? And I just mentioned that is you inject an animal with a specific antigen of interest, and then the body generates these antibodies, and then they are present in the serum. So we take a blood draw, we separate the serum, and the antibodies will be in that serum. So can you list some applications? I did list few at the beginning of the lecture. What did you capture? What are these assays useful for? Yeah, we can do it for protein purification actually, which is like affinity chromatography. Allergen testing, that's very good. What else did we get? Protein purification, allergy testing. Can you think of anything else? Very important applications. Testing pesticide, yes. Let me reveal some of. So important application is on the field looking for GMO. So that they use usually a test strip. We'll talk about the lateral flow test strip later on the field for testing GMO. Mixed species authentication. You want to determine adulteration in any case, like, okay, this is a beef burger, but it does have some horse meat. How are we going to determine the presence of horse meat in our beef burger is by immunoassays, where we have antigen specific for horse protein. Toxins like aflatoxins, pesticides, antibiotics, and also identification of bacteria and viruses. So very wide applications for immunoassays in food analysis. So we're going to learn ELISA today. We're going to talk about enzyme-linked immunosorbent assay. We have a very common form, which is the sandwich ELISA, which you might have heard of. There is another form, we call it comparative ELISA, and another form we call indirect ELISA. We're going to talk about each one of those. And then briefly, we're going to talk about immunoaffinity purification, lateral flow, test strip, and Western blot. So for ELISAs, we really need enzyme linked. So basically we need an enzyme, and the enzyme role here is basically to convert a substrate easily into a product that can absorb light. So that detection is measured by a spectrophotometer or basically a plate reader. We usually use 96 well plates so that we can run a lot of different samples, and then we can put that plate in a plate reader and get absorbance of the 96 wells simultaneously. It's a very neat technique. So the enzymes that are commonly used are easy to manufacture, fast-acting enzymes, and these are horseradish peroxidase and alkaline phosphatase. So these enzymes, usually one of them, is linked to an antibody or an antigen, depending on how the assay is. And then the antibody binds to the antigen, and then the enzyme is linked to one of them, and then you add a substrate, and then it converts to a product, and then you can quantitate that way. So this is the plate, the 96 well plate, and this is the plate reader. It's basically a spectrophotometer, but instead of having a place just to put your tube or your cuvette, you have a place for a plate that goes inside of the plate reader, and you can get absorbance measurements simultaneously for each singular well. So you get absorption in each of the wells, and the wells usually are numbered from 1 to 12, and then on the left here, A, B, C, D, E, F, G, H, and the total would be 96 individual wells. So the main steps, you have, first of all, you have your plastic surface, which is the solid hydrophobic surface. So here, I'm gonna draw it here. So that's the surface of your plate, let's say. It's hydrophobic. So you either coat it with an antigen or an antibody. And I'll show here an example where you put antibodies in the wells. And antibodies are proteins, remember? So they are going to bind via hydrophobic interactions. Sometimes they don't bind the way you want them to bind. They might bind upside down, and that is not very helpful, but at least a few will bind in such a manner where the binding sites are exposed. So the way we do that is we put our antibodies in the different wells, and we allow them time to bind to the surface, and then we do a wash. Oops, I'm trying to learn how to do this. So that's trying to write here. That's the wash, usually with a buffer or distilled water. So you wash off any non-reacting antibody that did not react. You just wash them off. And then what you have here is still empty surfaces. You don't want to put your antigen, and the antigen is a protein, let's say, if it's a protein, instead of interacting with the antibody, it might interact with the hydrophobic surface. So you don't want that to happen. So before you put your sample in the wells with the antigen, you put a blocking agent. And usually use a protein that you don't care for. It's not in your assay or in your sample. And we use either gelatin or bovine serum albumin. So this will bind here to these surfaces, blocking them from interacting, from your antigen from interacting with the surface. So that's we call blocking. All right, and then we do a wash. After every step, you do a wash. Next, you put your sample. Just gonna say sample. And then your antigen in the sample is going to bind. If you have antigen, it will bind. In sandwich ELISA, and you do wash after that. Anything that doesn't bind in your sample, different shapes of molecules get washed off. Okay, then you do wash. After the wash, you put another antibody. And this is, for example, in sandwich ELISA. We'll see other forms in a minute. It's called sandwich ELISA because the antigen is sandwiched between two antibodies. So, like I said, every antigen, especially if it's a protein, it has multiple epitopes. So one epitope is bound to this antibody and another epitope will bind to the other antibody that I would put. And thus it would be sandwiched. This antibody, the second antibody that I have, would be labeled with an enzyme. So an enzyme will be attached to it. I'll do a wash. I wash everything off. Then I'll add my substrate. The substrate will be added. The substrate will react with the enzyme and it will give me color. Okay, after every step, you do the washing. Okay, so I have my antibodies, my wash, blocking, I wash, sample, antigens bind, non-antigens are washed. I've then put my secondary antibodies, not secondary, I mean detection antibodies that are labeled with enzyme, and then I wash, substrate, and I wash, and then the substrate will react with the enzyme to give you a color, and then you measure absorption. In every step, you leave an incubation time for the reaction to happen. And that's how it is. This is important for you to know the label. I might give you a figure in the final and I say label that figure. So you want to know that you have your solid phase, which is basically the surface, hydrophobic surface of your plate. And then you have your capture antibody. So this is called capture antibody because this is the antibody that's going to capture the antigen from your sample. The red component here is your antigen. And then after that, you put the labeled antibody, which we call detection antibody. It's labeled with an enzyme. So then it binds to the antigen, forming the sandwich. The antigen is sandwiched now between two antibodies. And then you add your substrate, and then you have your colored product that you would measure. Read meaning measure. Okay, so that's the simple setup of the sandwich ELISA. Indirect ELISA is more common actually in assays that you would purchase in ELISA assays because they come with standard reagents. So for example, you can have monoclonal antibodies that come with the reagent kit that you would plate them in the plate. You would first have your capture antibody, could be monoclonal. And then you would have your, let's say if we're measuring an allergen, we get human sera to determine the immune reactivity of that protein. So I get human blood from a human that has allergy to that particular protein. So I get the antibodies here to react with the target protein. And it's called indirect. Why? Because this is a serum antibody that is not labeled. So, and it becomes really hard if we want to label it ourselves with the enzyme. It takes a lot more time in the lab and stuff. So it's always better to get the labeled antibodies already from a company that produces them. So we get antibody to the antibody. Does that make sense? So antibodies are protein. We can have antibodies that are produced in an animal, let's say a goat. We inject the goat with human antibodies, so then the goat produces antibodies to the antibodies. We take these antibodies, the company takes these antibodies and label them with horseradish peroxidase enzyme. And that becomes part of the assay. Already we can purchase that. That makes the assay easy because I can get the monoclonal from the company. I can get the labeled antibody to the antibody from the company, my substrate, everything. The only two things that I will have to worry about is the antigen, I mean the sample, and a human sera that has the antibody to that antigen. Everything else is pretty much in my kit. So that makes the indirect ELISA very common. Now both sandwich, like this is a sandwich ELISA, this is also a sandwich ELISA, but indirect. It's indirect because this antibody is labeled. The detection antibody here is labeled. Whereas in this case, this antibody is not labeled, but the antibody to the antibody here is labeled. That's the difference. But both, if you have more of your target protein in the sample, you get higher absorption in both cases, sandwich ELISA and indirect ELISA. If you have more antigen, you get more labeling bind, more enzymes to bind, and then more substrate to convert to enzyme, to product, sorry. All right, so with more antigen or more target protein, the higher is the absorption in both assays, sandwich ELISA and indirect ELISA. So these are just review questions for you. Comparative ELISA is another case. It's a different situation. Comparative ELISA is not meant for proteins. So if I ask you in the final to give an example for a comparative ELISA, if you say protein, that's going to be wrong. So just have to remember, comparative ELISA is meant for small molecular weight antigens. So molecules that are less than 5,000 grams per mole, or molecular weight of less than 5,000. So in this case, they are very small. So if you want to generate antibodies for that molecule, let's say aflatoxin or a pesticide, it's small. If we inject that directly in the animal body, it won't be recognized as an antigen. Antibodies will not be formed for this small molecule. It has to be linked to a bigger molecule. So in this case, that small molecule is linked to a protein. And when they are linked together, it is called a hapten. So this is the name, is a carrier protein linked to our antigen of interest. Because the antigen is too small, it can form only one epitope. It doesn't have multiple regions. The whole molecule is an epitope. So what happens here, if you look at this example for a comparative ELISA, you have the antibody, and if you have that antigen in your sample, that small molecule in your sample, it will bind, but it is very small. It cannot be labeled, or it cannot bind to another antibody. We cannot have a sandwich ELISA with it. So sandwich ELISA cannot be used for small molecule. Sandwich ELISA is mostly for proteins. So it cannot be used for small. So here, your antigen cannot bind to another antibody that is labeled. So how are we going to do the detection then? So in this case, we have similar antigen from outside of the sample. Let's say if we're looking at pesticide A in our sample, we get the standard pesticide A, and we label that with an enzyme. So what happens is when we put our sample and we put our labeled standard, what's going to happen, there'll be competition for binding. That's why it's called competitive ELISA. The labeled antigen and the antigen in the sample will be competing for binding sites. So the more I have of the antigen in the sample, the less labeled antigen will bind. And if I have less labeled antigen binding, I will get less conversion of substrate to product by the enzyme, and therefore I will get less absorption. So the lower the absorption I have, that means the higher antigen I have in my sample. And the higher absorption I get, that means I have less amount of the antigen in the sample and more of the standard labeled antigen with the enzyme bound to the antibody.
Can you repeat that one more time, please?
Yeah. So it's called comparative ELISA because the antigen from my sample is linked to the antibody here. And I have an antigen that is a standard. I I purchase it separately from my sample, and it is labeled with an enzyme. Okay, so what happens is I put my sample and I put the labeled standard. They will be competing to the binding sites on the antibodies. So if I have very low amount of antigen in my sample or zero amount of antigen in my sample, all of my standard will be binding to the antibody, and I get really high absorption because now I have enzyme on all sides, and enzyme is converting the substrate to product. If I have high amount of antigen in my sample, it will be competing with the labeled antigen, and then I'll have less absorption because I'll have less enzyme bound and I have less substrate converting to product. So in comparative ELISA, the higher amount of antigen I have in my sample, the lower is the absorption, and vice versa.
Thank you.
Yeah, so the label antigen binding produce the absorption because it is labeled with an enzyme. So when I put the substrate anesthesia, the substrate is converted to product with the enzyme that is present on the standard antigen that I added that is competing with the antigen in my sample. And that's what result in the reaction to produce the color. All right, so in comparative ELISA, you can have bound antibody or bound hapten assays. So bound antibody means I will have my surface with antibodies bound to it, and I will have a hapten that is labeled with the enzyme. Again, this would be a reagent that I add. So meaning a protein carrier with an antigen similar to the antigen in my sample, they're bound to form a hapten and labeled with an enzyme. So, and then this triangle here that is free, that is present in my sample. That's the antigen in my sample. So again, these two will be competing. The hapten labeled with an enzyme and the free antigen from my sample will be competing. Okay. So in the bound hapten, so what I have is the hapten bound to the surface. So this is a protein carrier. This is an antigen similar to the antigen in my sample. And in this case, the antibody is labeled with an enzyme. So the antibody labeled with the enzyme will bind to the free antigen if it's present in my sample and be washed off. Or if I don't have very many antigens in my sample, the antibody will bind to the hapten that is fixed on the plate. And then the enzyme will stay on the plate. And when I put the substrate, I'll get the color. Okay, again, competition. Here, the triangles that are free, you see those triangles. These triangles are from my sample. And the hapten is the reagent I put. It has similar antigen to my sample, and it has a carrier protein. So we bind that first, then we introduce the antibodies and the sample. And if I have more antigen in my sample, the antibodies that are labeled will bind to them and be washed. Whereas if I don't have very many antigens in my sample, the antibody that is labeled will bind to the hapten, will stick and convert a substrate to enzyme. One thing I want you to note here is that the hapten I use in my assay should be different. The carrier protein should be different than the carrier protein used in the animal to produce the antibody. Because then antibodies, instead of just binding to the antigen, they might start binding to the hapten itself, if it's the same protein that was injected in the animal. So it's very important that the hapten we use in the assay is different than the hapten used to produce that antibody in an animal to get that antibody in use in this assay. Okay. This is just to show you an application, for example, aflatoxin detection. So a sample that is high in aflatoxin, you will have those triangles representing aflatoxin binding with my antibodies and very little hapten with an enzyme or a substrate binds. So therefore I get less absorption. Whereas if I have 0 antigens in my sample, all of the labeled antibodies will bind and then I'll get a highest absorption, and that means I have no toxin. So moving on from ELISA, another application of immunoassay is immunoaffinity purification, which we talked about it in chromatography, where we have our stationary phase having ligands that are very specific to the compounds of interest. So sometimes the compound of interest are antibodies, so we have antigen bound. Sometimes we have antibodies on the column and a specific antigen that we want to purify or specific protein that we want to purify. So it's used for purification of specific protein or purification of a specific antibody. This is a common tool that you use, especially on the field when you don't have elaborate labs to run an ELISA or a Western blot. So use test strips. So they are lateral flow strips. The way it works, they're very cool. They are a form of sandwich ELISA. So this would be your strip. You would have immunobodies here in that blue spot here. That means immunobody labeled with a blue color. And here you have an immunobodies specific for the antigen of interest. So you dip your sample in your solution, and if you have the target antibody, it will bind to the color labeled antibody and move up by lateral flow. It moves up to this strip here where you have antibodies that are bound. So it captures. So they get captured and you see a blue line showing up here. So this is an actual strip where you have your sample pad, that's where the sample is placed, or you insert or you dip your strip in your sample. You have a test line and you have a control line. The control line is to make sure that there is no blockage and you actually have a lateral flow. So your color labeled antibody, if it finds the antigen, it will carry it to the test line where you have antibody to the antigen and it gets sandwiched and your antibody gives you the color. The rest of the antibodies keep moving up by lateral flow and they reach a control line that has antibody to the antibody, basically. So it captures the antibody and you see a color. So if you don't see a color in the control line, that means it's a faulty strip, you have to throw it away. So if you don't have antigen, if you don't have a target, what happens is your antibodies move, they don't get held here because you don't have an antigen, and then they go up to the control line and they get bound to the antibody and you get the color. So you always should get the color for the control. And this is basically the concept of the pregnancy tests that some of you might be familiar with or will be in the future. Okay, the last thing here is the Western blot. So the Western blot is basically uses an apparatus very similar to the gel electrophoresis. So you have a chamber where you put a running buffer and you hook it up to a voltage, current voltage source, and you run your sample. But the difference here is you already have gel that you've run. You either a 2D gel or a 1D gel that you've already run regular with SDS phage or a 2D with isoelectric focusing gel. So you have spots on your gel. You want to convert to transfer these spots onto a PVDF membrane, a polyvinyl difluoride membrane that it's a hydrophobic surface. So you want to transfer your proteins on top of that film or membrane. How do you do that is basically you need a compartment like this. So here this opens up, you have a white sponge if you can see here. When you open it up, you place your gel, so your gel layer here, and then you place your membrane here, and then you put your pad or the sponge, whatever you have, you put padding from the top and padding from the bottom, and you secure them here, and you place them in this chamber and you run a current and it goes, and the current goes and the proteins get transferred this direction. They go from your gel and the spots get transferred onto your membrane. So you end up with a membrane here with the different proteins. Now, you would incubate. So you incubate it with the primary antibody that binds to the antigen if it's there. Then you wash and incubate with the secondary antibody that is labeled, which is antibody to the antibody. It's an indirect ELISA in this case. So antibody to the antibody, it's already labeled with the enzyme. You add, you wash, then you incubate with the substrate. And then if you do have the antigen there, you will see a color developed at that spot. It's very cool. So this is an actual application where we're looking for a specific protein in pork meat. So you put a specific antigen that only reacts with that particular protein. So when we transfer the proteins, the only one that reacted and you see color is that particular band here in the pork meat lane. That is looking at authentication of the meat, checking for adulteration potentially. That's a great application for that. Also, it's a good screening method for allergenicity of proteins. You can screen, if you want to quantitate, you can do ELISA. Okay, that's it. We're almost time. Any questions? If not, have a great weekend, you guys, and I'll see you next week.